Thank you for joining us here for the Climate Research Minnesota presentation, which will be about 50 minutes in length. My name is Tanner Stoffren. I'm the Marketing and Communications Coordinator here at Wild Rivers Conservancy. Before we start, just a couple quick notes for everyone. On the right side of the screen, you'll see the chat window, and that is where, throughout the presentation, you can add questions and comments, which we'll address at the end of the presentation, and I'll make sure to sort through those. This session is being recorded and will be made permanently available on the Wild Rivers Conservancy YouTube account. With that housekeeping out of the way, I'd like to introduce our speaker and give the rest of the time to them. Please welcome Kara Baldwin of the Cedar Creek Ecosystem Science Reserve.
Awesome. Well, hello, everyone. I am the Education and Community Engagement Coordinator at Cedar Creek Ecosystem Science Reserve, and I'm really excited to talk to you about, one, Cedar Creek, but two, all of the interesting climate research that's happening here in Minnesota. At the very end of our time together, we will have an opportunity to dive into some of the data that we have collected and see what kind of patterns and things we can find along the way. So a quick orientation, we'll go to the next slide, a quick orientation to our day. If we get, there we go. We're gonna focus in on what is Cedar Creek Ecosystem Science Reserve, what we do at Cedar Creek. We're going to talk a little bit about Cedar Creek's research, and then finally we're going to explore some data from climate research. Go ahead and head on to the next slide. So I just realized that this we had to share my slides because they weren't sharing nicely, and so my apologies for all of the weird things that happened in the process of sharing. But this is, where is Cedar Creek? The thing to note is that we are about 45 minutes 35 miles north of Minneapolis, St. Paul. We are also, go ahead and head into the next slide. We're also an intersection of 3 different biomes. So one thing that makes Minnesota extra special and really fascinating is that we have 3 different biomes present within our state. We have the prairie or grasslands. We have the deciduous forests like oaks, maples, ash trees, things like that. And then we also have the coniferous forests like your pines and your spruces. And if you go ahead and click one more time through, you can see that here in Minnesota, we are in a location that can has those 3 different biomes intersecting and interacting with each other. Go ahead and go on to the next slide. So here, once again, as Minnesota zoomed in, these are those 3 biomes. The light tan color is the prairie, kind of the lighter green is the deciduous forest, and the darker green is the coniferous forest. If we click through to the next thing, you'll see that Cedar Creek, where it's located, is right where there's all these different places where these biomes are intersecting and kind of transitioning from one to another. So because at Cedar Creek we have all of these different biomes, it's a really great place to do science. So let's go ahead and skip on to the next slide. We have a lot of diversity at Cedar Creek, and we have examples of these biomes, and so we can study plants, animals, soils, other elements of the natural world, and really get a sense of an understanding of ecosystems, how they interact with each other, and as well as a nice big space to kind of manipulate what happens when human elements are introduced into ecosystems themselves. It's a fantastic place to do science. And so with that, we're going to scoot on forward. What do we do here at Cedar Creek? Cedar Creek is a field station owned by the University of Minnesota. We're connected to the College of Biological Sciences through the university. The science has been occurring here on site for over 80 years. And there are a bunch of groundbreaking science experiments that have happened here at Cedar Creek that have really transformed our understanding of ecology and how ecosystems work. But we're more than the science. We're a place to connect with nature. We have public hiking trails. We're a place for science and research. We're a place for learning. We do K-12 field trips and community programs. And then we're also a place with decades of history in the sciences. And we're going to go ahead and talk through all of those really famous science experiments and actually dig into some of the data of some of our science experiments with our time together today. So with that, we'll go ahead and scoot forward. Another thing that makes Cedar Creek special and unique is we are connected to the long-term ecological research sites. So this is something funded through the National Science Foundation. When you think about science, a lot of science is funded only for 2 or 3 years, and that's it. And so you can answer a lot of questions with funding for 2 or 3 years, but there's a lot of questions that you can't answer, especially when we're talking about ecosystems and climate change that needs a lot more time. And so because we're a long-term ecological research site, we have experiments that last for decades. 10, 20, 30 years of data, and looking at how that data is changing over time can help us really understand how to best deal with big problems like climate change. With the more data we have, the more accurate our conclusions can be, the more likely we're to see specific patterns or transitions in ecosystems. If you've ever thought about or heard about ecosystem transitions or succession before, you know that that can take a really long time, and we're able to see how climate may influence those things over time. And if we click it, you can kind of see there's going to be a little circle that pops up. And we're the little blue dot in that circle of all these other sites that are collecting similar data and doing other experiments looking at ecology long term. We're one of many sites throughout the United States. So knowing a little bit more about Cedar Creek, we're now going to talk about the science at Cedar Creek. And so scientists have been working at Cedar Creek for over 80 plus years, learning more about the habitats, the ecosystems, and things going on there. And so we're going to walk through some of the really famous science that's happened here. And the first really famous science experiment that I want to highlight is of Ray Lindemann. And so we'll go ahead and go to the next slide. Ray Lindemann, you can see his image there. Worked at Cedar Creek from the 1930s and the 1940s, and he and his wife Eleanor collected a lot of data. And basically, we understand food chains and how food chains work from herbivores and carnivores and back because of Ray Lindemann's work. Prior or most of the research kind of prior to this idea of how things interact in terms of food chains, a lot of it was worked on individual species. Ray Lindemann really focused in on how all of these individual species were interacting with each other in terms of the food webs, but also how energy flowed through those systems. So if you've heard phrases like trophic pyramids, things like that, that was really born out of Lindemann's work in 1942, and published work in 1942. So really famous science out at Cedar Bog Lake, looking at those food chain ecosystems. Going to the next slide. We also have a lot of research connected to wildlife movement and behavior. So radiotelemetry is something that scientists use to understand how animals move across the landscape. And at Cedar Creek, we back in the 1970s, 60s and 70s, they pioneered how to use radiotelemetry for wildlife movement and behavior research. So that was something that was really pioneered and engineered here in Minnesota at Cedar Creek. We also have continuing studies focused on thinking about how animals move around the landscape. We have a lot of tracking surveys and things looking at that. We have one of the larger breeding populations of the red-headed woodpeckers in Minnesota, and we do a lot of research and thinking about their behavior and movement through that project as well. And we also have a full series of trail cameras that tell us a little bit about where animals are. And so we capture all of these images of animals across our landscape. And then we can go and learn a little bit about what animals are using, which spaces, which habitats, which ecosystems within our property. And so one really cool thing is tomorrow, one of the options is for you to check out our Eyes on the Wild program, which is focused on wildlife movement and behavior. We'll go ahead into the next screen. Cedar Creek is also known for prescribed burning. We have some of the longest running prescribed burning experiments around, and they started in the 1980s, exploring how prescribed burning influences ecosystems. Fire is an important part of ecosystems like the oak savanna, which you can see pictured in the top right-hand corner there. And so we have various experiments looking at what happens or what's the burn frequency. And we've learned a lot about how prescribed burning can help maintain specific ecosystems through that research. We'll go ahead and go to the next one as well. We also have a lot of research related to climate change, thinking about how plant communities respond to things like carbon dioxide, nitrogen, temperature, and rainfall. And so we have specific rings. This is called our biocon experiment. These are specific rings of areas where we can artificially change the level of carbon dioxide going across plants to see how artificially increasing the amount of carbon dioxide influences plant communities. And this has been research that's been going on since the 1990s. And so we're able to start to see and kind of make predictions about what's going to happen with climate over time through some of this research. Go ahead and go to the next one. And the final research one I want to highlight is our big biodiversity experiment. This is the work of Dave Tillman, our director, and it really focuses in and is the first experiment to manipulate biodiversity, to experimentally change that, to get an understanding of how biodiversity influences plant communities, or whether or not it matters if plants are growing with their own species or with different species themselves. We have over 200 different research experiments or data experiments happening on site every year. So of course, this is just scratching the surface of all of the research and science that occurs and has occurred here at Cedar Creek. So many more topics and things that we explore related to ecology within our space. All right, we'll go ahead and head to the next slide. So for the rest of our time together, we're going to focus in on one very specific experiment, the biodiversity and climate experiment. This experiment is one that kind of came from other questions that we've learned from the past. So let's start with some definitions, and we're going to jump to the next slide. As you might expect from the name, this experiment explores two really big concepts, biodiversity and climate. So I think it's really good that we all have a shared understanding of what the scientists mean by those two terms and how we are understanding that in the context of this study. So the first is biodiversity. Go ahead and go to the next slide. Perfect. The word bio means life. And the word diversity means variety or difference. And so biodiversity is the variety of living things in an area. And biodiversity, if we go ahead and click one more time, biodiversity can be either very low. So this is one of the monocultures or single species plots through the big biodiversity experiment, or it can be a little bit higher. And so this is one of our 16 species plots at the biodiversity experiment. So you can see a very big difference in the level of biodiversity. In the next slide, we're going to explore, we know from the research of Dave Tillman and the Big Bio experiment that biodiversity does matter. Cedar Creek is a leader in the study of biodiversity. A lot of our research specifically looks at biodiversity. This big bio experiment was the first to manipulate biodiversity as the variable. And we know that having many different plant species together in one place helps plants grow faster. They can use water and nutrients more efficiently. They can resist disease and pests, and they can survive environmental disturbances like drought and function a lot better when there's more diversity than if, like, in one of those larger 16 species plots versus the single species plots. So biodiversity matters. We've learned that through research at Cedar Creek. So now let's think a little bit about climate. So climate is the average weather and seasonal patterns in a species over many years. Average weather and seasonal patterns in a specific place over many years. And so when we're thinking about climate, I always like to think it's about what you expect, and weather, in contrast, is what you would see in an individual day. So a nice way to think about this is in terms of closets. What you have in your closet might include things for winter, like sweaters, and snow boots and winter coats and hats and mittens. But you also have things to gear up for the summer, like shorts and t-shirts and swimsuits. You might have rain jackets and boots or lighter coats for the spring. So we think about climate, it's all those different clothes in your closet, the ones that it's kind of what you would expect that you would need living in Minnesota. If you were in Arizona or Hawaii, your closet would likely look very different in terms of the types of clothes you would need for the climate that's there. Weather is what you would put on every day from your closet. So weather is that individual kind of moment, what you get on a given day. Climate is the overall pattern over time. It's what you would expect. So now we have an understanding of climate. Let's think a little bit about what we know already about climate. Our climate is changing globally. And we'll go ahead and go to the next slide. So if we look over many, many years, it's really clear that our climate, or the average weather, is changing at a global level. You can see that the average air temperature on Earth has been getting warmer since 1980. And you can kind of see that pattern over time of that global average temperature moving upward. But we also know that our climate is changing at the state level. So if we go back to the next slide, we can look at some data related to climate in Minnesota. So these maps show how much our air temperature readings have changed in each season. If it is a red color, the more red it is, it means it's gotten warmer. If you see a blue, it's something that's gotten colder over time. Notice how pink and red each season is, especially in the winter. In some parts of Minnesota, it's now 4 degrees warmer in winter months than it used to be on average within our climate. You also can notice that there are some blue counties in the summertime. It is probably due to farmers growing corn and watering fields in the summer, and that added water in those particular regions acts like a body sweating and can cool off the temperature, the general air temperature around. But overall, the pattern is our climate is changing at a state level, and it is getting warmer, especially in the winter months. Go ahead and go to the next slide. And so when we think about Cedar Creek, we started collecting climate data at Cedar Creek in the 1930s. We have noticed that our climate is changing at Cedar Creek as well. And if you, so this is a great picture from 1937 of one of our sites, Cedar Bog Lake at Cedar Creek. And if you go ahead and click to the next thing, since the 1940s, we've seen an average minimum temperatures have increased by 2 degrees Celsius, and the average daily maximum temperatures have increased by 1.2 degrees Celsius. And so we have already seen the average daily temperature go up by more than a degree. So now that we know that it's changing, how much warming can we expect to see in the future? Globally, we are depending on what choices humans make, and we all have a choice to make a difference here. Where we get our food, how we use our energy, how we manage environments. We're probably going to see the temperature increase 2 to 5 degrees Celsius, or a little bit more by the end of the century. When we talk more locally, like at Cedar Creek, we're really looking at a similar scenario. Scientists think that we'll see that similar change in temperature, and this can have really huge impacts on the things that we measure in our ecosystems, like how much water is available in soil, or when will the plants bloom, or how many plants are growing each year. And even decomposition can be influenced by this changing climate. So knowing all of this information, the Biodiversity and Climate Research Project really wanted to think about how does initial conditions like biodiversity influence the nature and magnitude of a warming climate. If we can go to the next slide. Oh my goodness, I'm so sorry that this did not translate well. So biodiversity will influence the nature and magnitude of a warming climate. But we really wanted to explore in what ways. How does the initial conditions like biodiversity level influence the warming climate? And so we don't know some of that information. And so that's where we can learn a little more. So the Biodiversity and Climate Experiment is really focused on trying to answer that question. So what specifically did they do, and how are they exploring this question? If we go to the next slide, we have 30 from our Big Bio experiment. There were 32 randomly selected plots with varying biodiversity levels of either 1, 4, or 16 prairie plant species. And so you can see the big plots with all of the different squares, 9 by 9 meter squares of different biodiversity levels in our fields. In the Big Bio experiment, and then a handful of them were chosen based on the number of species present in each plot. And so you can see the 1 and the 4, you can see the single species plot, and then I believe that is a 16 species plot, excuse me, in the upper right-hand corner. We have already can kind of tell, right, which ones are a low biodiversity and which ones are a high biodiversity. But if you look really carefully at that larger picture of the overhead picture, the drone picture, you can definitely hopefully notice some of the differences even from an aerial view between the different plots, which ones have low biodiversity and which ones have higher biodiversity in the kind of peak of the growing season. So we have these varying biodiversity levels, and we also have 3 different treatments per plot. First, we have our control plot, where there's no warming at all. We have our low temperature area, where we're increasing the temperature by 1.5 degrees Celsius. And then we have a high temperature increase of 3 degrees Celsius. And again, I apologize for the placement. It seems like in the sharing of this, all of the pictures got out of place, and so they're difficult to see. What you can see in the picture is how we change the warming amounts. Those little kind of tent looking things above the plots are heaters, and so those heaters are calibrated to increase those temperatures just a little bit or a little bit more. And then we have one of them that's always off. That's our control, to take into account how like shading might influence things, or kind of to make sure that we're not missing something. So we just have one that's just the tent, and then 2 of the tents with heaters. And they're turned on from March until November. So they have a pretty long time that these lamps are turned on over the course of the growing season. So I'm going to ask a quick question. What do the scientists manipulate? And so then what are the independent variables? So what are the two independent variables in the biodiversity and climate experiment? And I'll have maybe a couple answers come into the chat, and then we'll talk about it a little bit more. And I'm going to take a quick drink of water to between. All right. Well, we'll go ahead and, oh, the heat. Awesome. Yep. So the heat is one of the independent variables, and the other is biodiversity. So we'll go ahead and click through so you can see those two different variables. The diversity, or how many plants are growing together in a plot, and then the warming treatment, either control, low, or high warming. But scientists also need to then measure things, or have our dependent variables. And so we have, what do scientists measure? The big thing that we're exploring here, and I'm so sorry that this is not translating well, is the productivity. In this particular research, productivity is measured through above ground biomass. And biomass is just a big fancy term for basically how much plants grow each year above the ground in each plot. So these are perennial plants. So every winter, they're kind of the stuff that grew last year will die off, but then the new stuff will grow every year. And so they're really measuring how much growth occurred in each plot over time. That's the primary variable that's collected, but we need to think about other things too. There are things like below ground biomass. Below ground biomass is how much root kind of structure is going on, what's happening under the ground. We have soil carbon and nitrogen, which is the amount of carbon and nitrogen stored in each of in the soil in each of the plots. We have phenological changes like budding time and growth rate. Phenology is like the study of seasons, or the study of timing. And so how quickly, or when do flowers bloom? How long do they bloom for? Things like that. And then also looking at the soil microbial and community dynamics. And so those a few more bullets to kind of highlight and click through really quickly of all those things we talked about. So that below ground biomass, the soil carbon and nitrogen, the phenological changes, and that soil microbial community dynamics. So those are all the dependent variables that were measured. But scientists also need to measure other variables that might help explain some of the patterns that are seen. And so in this experiment, they also measured vapor pressure deficit, which is a complex idea, but the big takeaway is it's how well plants are able to hold on to moisture. And if it's really easy to hold on to your moisture, you have more water available to do the things you need to do as a plant, to grow, to photosynthesize, to create energy, things like that. If it's hard for you to get water, then it's a little more tricky as a plant. And so it's that measure of how well basically plants can hold on to their water. That's a really simplified way of thinking about vapor pressure. If we go back one really quick, the other thing is the light availability at ground level. So that's just measuring how well does sunlight travel all the way to the ground within the plots themselves. So those were the two variables that were also explored to think about how these patterns might be there. And we can ask a lot of questions from that, right? Is water evaporating out of the soil? Is there more bare ground? What kinds of things could we explore more fully? So scientists then can ask a bunch of different questions related to the variables this specific experiment. So for example, we can look and think about, is there an impact of warming on the plant productivity from year to year, or how does that change from year to year? Within a diversity level like 1, 4, or 16, how does warming impact productivity? And finally, can vapor pressure deficit help explain the different patterns that we see? Are some of the questions that scientists have asked over the time. And so we'll see how this works, because so far I've been unable to share my screen. But we have, and I've shared with you all in the through the community kind of portal, some of the raw data from the experiment. And I wanted to walk through that data with you and explore some of these patterns and what we might see. I don't think the screen sharing issue has been resolved, so I'm hoping Tanner is up for a crash course in how to use to walk through some of this. If you went and click one more time, Tanner, there are a bunch of different pieces of data in the data set available. We have the year, we have the diversity level, and warming treatments. We have the above ground biomass, the vapor pressure deficit. And so you'll be able to analyze the data set and kind of graph some of your own results through either through the spreadsheets that I've provided. And I think for the time that we have left, we'll be exploring that data and seeing what kinds of things and patterns that we can see, and really think about this raw data like a scientist would. And so we'll go ahead and switch over to the data set and explore it in our through the rest of our time together. All right. And I just lost the group. There we go. So here is the data set. This is the raw data. And you can see across the top. So I'm going to kind of go down and help you all out in understanding what we're seeing here, and then and explore how we can analyze this data. So the first kind of part of the data is plot. Plot literally is the number of plot. If you remember that kind of quilt like looking shot over the top of drone shot over the top of big bio experiment, each one of those squares has a very specific number. That is the plot or the data that we have for each of those big bio things. So plot number is there. Year was the year the data was collected. In this particular spreadsheet, we have the data from 2009 to 2015. Diversity is that independent variable that was manipulated, that was changed. So these were plots that were planted in 1993. So they have had a long time of growth and establishment. There's diversity plots of 1 species, 4 species, or 16 species. The next one over is the warming treatment. That's the other independent variable that was explored. We have our control, we have our low, and then we have our high temperatures collected within each of those plots. Then we have our productivity. So the productivity values and numbers are the ones related to how much biomass grew, or how much the plant grew over the course of the growing year. So how much stuff the plants added above the ground. It's measured in grams per square meter. Then the next one over, we have the vapor pressure. So this is kind of a measure of dryness from the perspective of a plant. The actual measurement is the difference between the amount of moisture in the air and how much moisture the air can hold when it is saturated at a particular temperature. And so a higher values mean the air is drier, and the growing conditions are poorer. So it's important to remember that vapor, like the productivity, larger is better. When we're talking about vapor pressure, lower is better. And then the final one is that light, percent light transmission. And that is the idea of how much light is actually hitting the ground at the base of the plots themselves. So we have all of this data to work with. Some of these variables are categorical. Actually, only one of these variables is categorical. It is the warming condition. Categorical just means it's either kind of put into these three categories. The rest of them are quantitative or numerical values. And we can then start to manipulate this data and explore some of these quantitative variables to get an understanding of how biodiversity and climate change might interact. And so if we scoot over to the next tab on the spreadsheet, I just reorganized the data to make it nice and easy to explore some of these different kind of quantitative variables. So for this first little bit of time together, I want to, highlight that a pattern. So let's go ahead and highlight diversity. And just so click the C at the very, very top, and then hit Command or Control and hit the E at the very top. And then if we want to insert and then a scatter graph. If it'll let us do it. My plan was that I would be doing this live with you. And it might not allow us to edit. Anyway, we can play and explore this data and manipulate it to try to, not manipulate it, we don't change the data, but we can look and see and graph these things to see what kind of patterns exist. And if it's not working right now, that's okay. You all have access to this data and can start to interact and consider how these different things might interact. The other thing I wanted to highlight, and I'm going to type it in the chat, is we have this data through an interactive tool made by one of the primary project scientists, Dr. Jane Cowles-Dittmer, to kind of look at this data through a more maybe if you're less familiar with Excel, this might be a more user-friendly way to explore the data. And if you go to that particular website, oh, awesome! Thank you for getting the two quantitative variable pattern going on. So you can see that there is an increase in the data. There's this slight increase in terms of the productivity between the different plot types. So this is biodiversity and productivity. This is in line of what we already know from the big biodiversity experiment, that productivity will increase over time due to the that biodiversity increases plant productivity over time. So that's one way you can explore this data, is by comparing all of these different components and how they interact with each other. The other place to go, and Tanner, if you wouldn't mind just jumping into the Z link, I just want to highlight this really cool resource as well. And this Z link is the same data set, this exact data set that you can kind of explore. You can also have how the scientists explored and looked at this data to see what was happening. So you can click on the different things to see how diversity changed the various and how diversity and climate change interact. And basically, when we're talking about primary production or how much biomass is above ground, we're finding that diversity plays an impact in how much of that primary production is occurring. And so as it gets warmer, there's more primary production in the more diverse plots than the less diverse plots. And if you scroll down a little bit on that website, it's part of the reason, or the way the scientists have been thinking about this, is the idea of the plant biomass production. And so, and the water vapor deficit. And so you can see that there are, as you switch between the different plots, you can see kind of what the plots look like, but what's happening in terms of the how it's the more diverse plots are able to hold on to more of the water. There's less evaporation of that water. And so they're able to then provide and have all the things they need to grow and produce and get kind of increase how much growth that happens over that growing season. So lots of fun things to explore regarding the data. I apologize that the screen share wasn't working very well on my end, and I appreciate you all going with the flow with me today. I do want to provide a little bit of time for questions if anyone has any. If not, it was a pleasure presenting to you all today, and I look forward to you reaching out if you have any questions or want to learn more about climate science in Minnesota.
And Tanner here.
Tanner.
Yeah, thank you for collaboratively working through that with me on the fly. I have one. Over the course of the year, how often are you measuring those plots? Is it a daily basis, or how frequently are you collecting that data?
So the biomass is one clipping at a very specific moment in time, so that we have it the same basic time every year. And so that comes in with the long-term thing. And so basically we're getting a sense of the biomass growth from the season, and then we can compare season to season to season, year to year with that clipping. I think it helps sometimes understand like how we actually measure it. So we have to clip all of the, not everything, but they have to clip some of the plants down. They collect every little blade of grass, everything that's been clipped, and it gets dried so that they remove the water. And then it also has to get sorted. So they have to sort each thing by individual species based on, is this blade of grass this species or this species? And so there's a lot of work that goes into how this data is organized. But yeah, it's very much focused season to season. So the biomass itself is only measured that one time, once a season.
Awesome.
And then the analysis takes a while.
I'm sure. A lot of time after that. We have one from Katrina. Is this research something that is going to continue for a long time going forward?
So this research, specifically the biodiversity and climate change research. So the biodiversity experiment has been happening since 1995. I think they started collecting data. It started in 1993. This specific experiment started, I believe, in 2009 with the heat differential treatments. And it has been collected since. So this is an ongoing experiment. I don't know how much longer it will continue, but as a long-term ecological research site, we do have the funding to allow us to do research for long periods of time to look for what kinds of patterns are we seeing. And we're noticing that with climate change, some of these patterns are taking 10, 20, 30 years before we see like community turnover and things like that. So it's really important to have those long-term data sets so that we can really get an understanding of how climate and human influences can really change ecosystems, not only in short term, but in the long term.
Awesome. October asks, what are some experiments we can do in our own backyards to show us the effects of climate change in our individual microclimates?
Oh, very interesting. I think one of my favorite things to think about in terms of the backyard is really focusing in on phenology. I've used that term a few times, and if you're not familiar with it, there are whole like phenology networks out there that you can do some like Googling on and everything. Phenology is kind of the study of change over time in terms of seasonality. And so it's like, when do the buds burst? When do we see our first robin? When do you notice, the first snow? What is happening in all these things? And so I think one really cool thing you can do in terms of a long-term backyard experiment is to really think about how does the seasons, or how does different kind of key things connected to season change over time, or when does it happen? So like if you have a favorite spring flower, like when are the tulips blooming, or when are the lilacs blooming? I know those aren't necessarily natives, but and then marking those dates and doing that year after year to see if you're noticing any patterns. So I think that's a pretty fun way to do it, but it is looking more long-term than short-term for sure.
Well, time for one last question. Continuing that theme, what combinations of plants sequester the most amount of carbon from the atmosphere?
So the answer is diversity is really important. So in our plots, we have C3 grasses, C4 grasses, forbs, and legumes. And they're finding that those four interacting with each other are really important. What's really interesting about that is diversity is what will do that. So the plants kind of, when they're all those different types and all those different functional groups, some of them are going to have a really good year, and some of them might have a bad year depending on the weather conditions. But when they're all together, they kind of almost all help each other grow. Like some of the plants hold in more potassium and provide more potassium for the environment. Some of our grasses are really good at holding in the carbon. Our legumes are good at providing nitrogen for the other plants to grow. And so they all are interacting with each other. So I think the answer to that question is diversity of 16 species planted near each other. I think would be at least 16 species planted near and around each other would be a really good way to think about that, because that is what's providing that growth, that production above ground. And I will say, like, every way that we measure diversity matters. So if it's production above ground, if it's the roots below ground, if it's resistance to disease, every way we measure it, it just seems like adding more plants, more different types of plants is really, really important in terms of the health of the ecosystems themselves.
Awesome. Excellent.
Yeah.
Well, I'd like to thank you for your awesome presentation. It's informative and great to follow and see how everything is unfolding in our local ecosystem, and the hard work and measuring that you do to stay on top of that. I'd also like to take the last couple minutes here to thank everybody for joining us today for the 2023 St. Croix Youth Summit. This presentation and all the others will be shared on YouTube again on our Wild Rivers Conservancy account. Email link will be sent to all of you so you can access that, as well as you will receive a survey. So please take a few moments to fill that out and let us know what you thought of today. That's appreciated. So thank you, Kara. Appreciate it.
No, you're welcome.